Welcome to the Breakthrough Broadcast, a podcast about innovative STEM solutions to real-world problems. From emerging technologies to practical research, Drew Reck explores how science, engineering, and bold thinking are shaping the future.
Hi, I'm Drew, and welcome back to the Breakthrough Broadcast. Last episode, we researched and learned a little bit about CRISPR, the gene editing technology that has completely reshaped biology. We started with its discovery, how scientists uncovered a natural bacterial immune system capable of remembering viral invaders, and how Jennifer Dunda and Emmanuel Charpentier harnessed this system to create CRISPR-Cas9, a tool precise enough to target any DNA sequence. We explored the molecular mechanics, how Cas9 acts as a molecular scissors guided by RNA, cutting and allowing cells to repair individual DNA, either with small errors or by inserting new genetic material through templates. We also looked at how CRISPR is being applied today. In medicine, it's already showing promise in curing monogenetic disorders like sickle cell disease and certain forms of inherited blindness. We examined its role in engineering immune cells to fight cancer, and even in basic research, where it accelerates the creation of model organisms to study disease. Beyond medicine, we saw its impact in agriculture, from creating crops that resist drought and disease to experimenting with livestock for improved traits, and in industrial and environmental biotechnology, where microbes are being programmed to produce chemicals or absorb carbon. Importantly, we didn't shy away from the challenges. CRISPR is imperfect. Off-target effects remain a concern, especially in human therapies, where unintended edits could have serious consequences. High-fidelity Cas9 proteins and careful RNA design help, but they come with trade-offs in efficiency and cost. Today, in part 2, we're shifting our focus from what CRISPR can already do, to what it might do next, including the completely new topic of ecosystem-level engineering. We'll explore the potential for programming soil microbes to enhance crop growth, restore degraded lands, and even combat climate change. We'll examine the scientific possibilities, the technological limitations, and the ethical questions that arise when humans start engineering life on a larger scale. By the end, you'll understand not just the power of this next frontier, but also the responsibility that comes with wielding that power. Like I just said, the first part of this episode is about the future of the technology, as we already have the knowledge from the previous episode about what the tech is and what it can do. We have a base for learning more about how the technology can mature and grow into its full potential. Looking ahead, CRISPR's potential stretches far beyond what we've already seen in medicine and agriculture. One major potential area of expansion is the treatment of complex diseases. Whereas monogenetic disorders, or disorders that are only caused by one DNA mutation, like sickle cell disease, are already being targeted, companies such as Verve Therapeutics are exploring CRISPR to address polygenetic conditions, such as high cholesterol and heart disease. Their approach is to edit genes in the liver that influence lipid metabolism, aiming to reduce lifelong cardiovascular risk with a single treatment. Similarly, Beam Therapeutics is developing base editing, a next-generation CRISPR technology that allows scientists to change single DNA letters without cutting the DNA strand, offering unprecedented precision for correcting mutations that contribute to multifactorial diseases like type 2 diabetes or some autoimmune disorders. Another frontier lies in infectious disease prevention. Researchers are investigating how CRISPR could be used to create universal and antiviral therapies. For example, some companies, including Locus Bioscientists, are designing CRISPR-based antimicrobials that can selectively target antibiotic-resistant bacteria. Unlike traditional antibiotics that attack both harmful and beneficial microbes, CRISPR-guided nucleases can hone in on specific pathogenetic DNA sequences, potentially in eliminating dangerous bacteria while leaving the microbiome intact. In the viral space, scientists are exploring CRISPR-based antivirals that could target DNA or RNA viruses in human cells, potentially providing rapid responses to outbreaks like influenza or even emerging viruses that can trigger global health issues. This is even more valuable than you might think, because current infectious diseases have a trend of growing resistances to the current antibiotics. This is to the point that superbugs are developing, or infections that have evolved resistance to all of the current antibiotics we have, meaning that without new antibiotics and infection-fighting strategies, like CRISPR can be, we have no tools for defeating them. This means that CRISPR is incredibly valuable in defeating these superbugs. Agriculture is another domain poised for radical transformation. Beyond drought resistance or pest tolerance, Future CRISPR applications could create crops with entirely new capabilities. As we said in the last episode, scientists are already experimenting with plants that produce novel compounds, like enhanced vitamins, pharmaceuticals, or industrial enzymes, directly in the plant tissue. For example, edible vaccines could be grown in rice or potatoes, allowing populations in remote regions to gain immunity through food rather than injections. Companies like Pairwise Plants are working on accelerating plant breeding using CRISPR, enabling the creation of traits that would normally take decades to emerge naturally. Beyond individual crops, CRISPR may enable designer ecosystems, where multiple plant species are being edited to coexist symbiotically, improving soil health and carbon sequestration while boosting yields, a completely new approach to sustainable agriculture. Industrial biotechnology is another field with potential breakthroughs possible. CRISPR can be used to engineer microorganisms that produce biofuels more efficiently than traditional methods. For instance, Ginkgo Bioworks is designing microbes to synthesize chemicals, flavors, and materials in ways that reduce waste and energy consumption. In addition to making new processes, this technology can also make old processes more efficient. Beyond the production aspect, CRISPR is also making breakthroughs in the environmental arena. CRISPR is being explored to engineer microbes that can degrade plastics, clean up oil spills, or remove greenhouse gases like methane and CO2 from the atmosphere. These applications go beyond replacing current methods. They open entirely new ways to address environmental problems at scale. Even in human enhancement and longevity research, CRISPR has the potential to innovate. Startups are investigating whether gene editing can improve cardiac health in aging dogs. While this might sound silly, it is meant to be a stepping stone toward producing treatments for poor cardiac health in humans. Other speculative applications include enhancing muscle function, improving resistance to disease, or even editing the human microbiome to influence metabolism or immunity. While these remain experimental, they illustrate the broad imaginative scope of CRISPR's future. Beyond these applications, the evolution of CRISPR itself is accelerating. New tools such as prime editing, a new method that is more accurate and consistent than traditional CRISPR, are allowing scientists to tackle previously undruggable mutations. Combined with artificial intelligence, these technologies could allow the design of edits that optimize health, increase crop productivity, or engineer microbes for entirely new chemical reactions. AI can predict off-target effects, design optimal guide RNAs, and simulate genetic networks before any experiment is performed, making CRISPR applications safer and faster to develop. Companies like Inscripta are working on automating CRISPR's workflows, enabling large-scale, high-throughput experiments that could produce new medicines or materials at a speed unimaginable just a few years ago. CRISPR is also poised to impact global food security in ways that extend beyond single crops. Scientists are experimenting with creating plants that can grow in saline soils or that fix more nitrogen, reducing the need for chemical fertilizers. This means more efficient crops and more plentiful crops. Future applications could include developing entire regions of land for cultivation that were previously unsuitable, potentially addressing global hunger and malnutrition. In addition, CRISPR could be used to produce crops with optimized nutritional content, tailoring vitamins, antioxidants, or essential amino acids to meet the dietary needs of specific populations. In animal biotechnology, the next generation of CRISPR interventions goes beyond disease resistance. Researchers are envisioning livestock that produce less methane, reducing the carbon footprint of farming, or that have improved welfare traits, like enhancing immunity or decreased susceptibility to heat stress. Some startups, like Recombine, are exploring ways to combine CRISPR with synthetic biology to accelerate these changes in farmed animals while ensuring precision and safety. Beyond traditional livestock, CRISPR could also influence aquaculture, creating fish or shellfish that grow faster, resist disease, or tolerate changing ocean conditions. Finally, CRISPR's potential extends into entirely new scientific frontiers. For example, gene drives. CRISPR-based systems can rapidly spread a genetic change to a population, could theoretically control invasive species, or prevent the spread of diseases like malaria by editing mosquito populations. In synthetic biology, CRISPR could be used to program cells that perform specific tasks, such as producing pharmaceuticals on demand, sensing environmental toxins, or assembling nanomaterials. Even in space biology, CRISPR might be used to engineer microbes that can help sustain life on long-term missions, recycle waste, or produce oxygen and nutrients in closed ecosystems. In short, the future of CRISPR is exceptionally vast and deeply practical. It encompasses applications in human health, agriculture, industrial production, environmental management, and even speculative science. Companies and research teams around the world are not only refining the technology itself, but also exploring innovative ways to deploy it, ways that could solve problems we haven't even considered yet. Unlike the current wave of CRISPR applications, which mainly improve existing treatments or crops, these emerging uses promise to create entirely new solutions, fundamentally altering medicine, agriculture, industry, and our relationship with the natural world. By leveraging advancements like base editing, prime editing, AI-driven design, and large-scale automation, CRISPR's next chapter will likely redefine what is scientifically and socially possible, setting the stage for a future where genetics can be guided with unprecedented precision and impact. I've just spent the last 10 minutes just talking about how great this tech is, how fast it's improving, its potential, and its practicality. All of these things are true, but considering only that angle is closed-minded and incorrect. We also need to consider the things that are keeping us from achieving that future. One of the most persistent technical hurdles is delivery. CRISPR components, Cas9 and guide RNA, must be transported into the correct cells efficiently and safely. Current approaches include viral vectors, lipid nanoparticles, and electroporation, each with strengths and weaknesses. Viral vectors, for instance, are naturally adept at inserting genetic material, but can trigger immune responses and sometimes integrate DNA in unintended locations, causing mutations. Lipid nanoparticles are safer, but often struggle to reach certain tissues like the brain or internal organs. However, this limitation is not as big of a problem as the issue with viral vectors, and so they are the main type of CRISPR used currently. Electroporation, which uses electrical pulses to temporarily open pores in cell membranes, is effective for ex vivo applications, meaning only have the opportunity to take out and edit cells outside the body, but it cannot be applied safely to living patients directly. Each method requires careful optimization, and no universal solution yet exists, making delivery one of the central challenges in translating CRISPR from lab experiments to widespread therapeutic use. Off-target effects remain another critical concern. Even with carefully designed guide RNAs and high-fidelity Cas9 proteins, unintended cuts, and therefore disruption of the DNA chain, can occur. These mistakes can disrupt unrelated genes, activate oncogenes, or interfere with regulatory sequences, producing consequences that may be unpredictable or dangerous. In medical applications, such as editing human stem cells, off-target mutations could silently propagate as cells divide, potentially causing cancer, immune disorders, or other complications. Agricultural applications are not immune to these off-target effects either. A single unintended edit in a crop could reduce yield, increase susceptibility to disease, or alter nutritional content, effects that might not appear until several generations later after a farmer's yield has decreased for a decade. High-fidelity Cas9 variants reduce off-target activity, but are slower, more expensive, and less efficient, creating a trade-off between precision, cost, and time. For large-scale applications, these limitations are significant, influencing which projects are feasible and which remain experimental and don't receive funding. Polygenic disorders highlight another layer of difficulty. While monogenetic diseases caused by a single defective gene can sometimes be treated effectively with CRISPR, disorders involving multiple genes still remain out of reach. Even when targeting a single gene, the cellular repair process introduces complexity. After Cas9 cuts DNA, this cell repair must happen. Two main pathways exist: non-homologous end joining, or NHEJ, which is fast but error-prone, and homology-directed repair, which is accurate but inefficient and only active in certain cell cycle stages. Scientists can attempt to bias cells toward HDR, or homology-directed repair, using chemical treatments or engineered proteins, but these interventions add cost, complexity, and potential risk. The precision of editing is not simply about cutting DNA, it is about controlling how the cell fixes itself afterward, a step that remains challenging and is somewhat unpredictable. Immune responses further complicate CRISPR's application. Many human cells have pre-existing immunity to Cas9 proteins derived from common bacteria, meaning the body may attack or neutralize the machinery before it can complete its work. This not only reduces efficiency, but can cause inflammation or tissue damage. Solving this requires protein engineering, transient delivery methods, or immune suppression, each of which adds additional layers of complexity, risk, ethical concern, and cost to operations. Technical limitations of CRISPR intersect closely with profound ethical questions. The ability to rewrite genes in humans, animals, and plants raises concerns about safety, consent, and unforeseen consequences. Editing somatic cells, those not passed to offspring, generally carries fewer ethical implications, since the changes only affect the treated individual. Germline edits, however, which alter DNA in eggs, sperm, or embryos, are far more consequential, potentially introducing permanent changes into the human population. Even minor errors could propagate unpredictably, creating new diseases or traits. Most countries currently restrict germline editing, reflecting a consensus that moral judgment must not lag behind technical capability. Central debate revolves around whether humans should be able to influence traits in their children. Possibilities like selecting gender, enhancing intelligence, improving physical abilities, or reducing disease predispositions raise complex moral questions. Religious perspectives sometimes view such innovations as interfering with God's design, while others worry about the disruption of natural evolutionary processes, reduced genetic diversity, or societal pressures favoring certain traits. Critics also caution that prioritizing enhancements could amplify social inequality or create unrealistic expectations for parents and children alike. Counterarguments note that humans have influenced evolution for millennia through agriculture, selective breeding, and modern medicine. Plants, animals, and even humans have been indirectly shaped by interventions such as vaccines and antibiotics, which have altered survival and reproduction patterns. From this perspective, deliberate, precise genetic editing could be seen as a continuation of humanity's long-standing influence on biology, albeit now with far greater control and responsibility. Intent and scope are also critical ethical factors. Editing genes to prevent debilitating diseases such as muscular dystrophy or hereditary blindness is widely considered morally justifiable, since it alleviates suffering without modifying non-disease traits. In contrast, interventions aimed solely at enhancement, intellectual, athletic, or aesthetic, raise broader societal concerns. Beyond equity, they could intensify competition among parents and perpetuate harmful ideals of perfection. The long-term consequences for individuals, families, and society remain unknown, requiring careful deliberation and regulation. Ethical concerns also extend to environmental and agriculture applications. Gene-edited crops or livestock can offer higher yields, better disease resistance, or increased efficiency, yet their deployment must consider ecological consequences, biodiversity, and the livelihoods of farmers. Technologies like gene drives, designed to propagate traits through wild populations, carry potential for cascading ecological impacts if mistakes occur. Ensuring accessibility is equally important. Cutting-edge therapies are only available to wealthy populations. Social inequalities could worsen. Regulatory oversight and public engagement are essential. International committees, ethical boards, and government agencies are establishing guidelines to ensure that CRISPR applications are safe, transparent, and socially responsible. Ethical frameworks must evolve alongside scientific innovation. Public dialogue and education are as critical as laboratory research. Balancing technological feasibility with moral responsibility will ultimately shape how society harnesses the power of gene editing. In summary, CRISPR is a transformative technology constrained not only by scientific limitations, delivery changes, off-target effects, polygenetic complexity, repair pathways, unpredictability, and immune responses, but also by ethical considerations surrounding safety, equity, ecological impact, and human enhancement. These intertwined concerns underscore that CRISPR is not a tool to wield without deliberation. The future of gene editing depends as much on thoughtful regulation and public engagement as it does on laboratory breakthroughs. Addressing both technical and ethical limitations will determine the pace and direction of progress, defining how humanity navigates the profound responsibility of rewriting the code of life. As we wrap up, it's worth stepping back and reflecting on the broader significance of CRISPR. This technology represents more than a set of molecular tools. It embodies the potential of human ingenuity to address problems once thought unsolvable. From accelerating medical breakthroughs to transforming agriculture and industrial processes, CRISPR demonstrates how precise scientific insight can be applied toward real-world challenges. Beyond the lab, it offers a lens into the future of innovation, showing what is possible when curiosity, creativity, and interdisciplinary collaboration come together. Looking ahead, the impact of CRISPR will extend not only to scientific progress, but to society itself. It encourages new ways of thinking about sustainability, global health, and the interconnections between humans and the environment. The stories of companies, researchers, and communities embracing this technology illustrate a shared vision: harnessing knowledge and responsibility to create meaningful change. While challenges remain, the unfolding potential is enormous, inspiring optimism about what can be achieved when science is paired with thoughtful ambition. Ultimately, CRISPR invites us to imagine a world where the boundaries of biology are no longer fixed, a world in which innovation can improve lives, strengthen ecosystems, and expand the horizons of possibility. The future of gene editing is just the beginning, and how we choose to shape it will define the next era of human achievement.
Thanks for tuning in to the Breakthrough Broadcast. If today's conversation sparked a new idea, share the episode and follow the show wherever you listen. Got a question, idea, or breakthrough of your own? Reach out at breakthroughbroadcastconnect@gmail.com. Until next time, keep questioning, keep building, and don't wait for the future. Help create it.